And that was it, or? Okay, any other questions? Okay, so if not, we're going to switch to the more workshop specific part. And essentially where this is an outline of the workshop. And so we're going to start with raw reads coming out of the sequencer. So which is essentially the way you normally get this is, I mean, you get in touch with the sequencing core, and you end up agreeing on like a specific sequencing approach. And then you send them your biological material. And then at some point, you're going to get an email from the core saying, hey, we've sequenced your sample, and here's a link to download the data. And those data, they're going to be FASTQ files. We're going to see this in a second. And so you're going to get raw reads, and they're just like files with sequences and sequences and sequences for small file. We're going to see this in a second. And then there's going to be a number of steps. Essentially, we're going to work on raw reads today, and then tomorrow we're going to work on aligning reads to a reference genome, and then on Thursday we're going to work on quantifying gene expressions based on these alignments. And so the end goal is going to be arriving to this just quantification of per gene expression in all our samples. And that's going to take the form of a table like this, where we might have like 3 samples. And then for each sample, we're going to have an expression value for each gene. And that expression value is just going to be the number of reads that map to that particular gene. Okay. And so, yeah, so today we're focusing on just the raw reads. And so this is a Sanger sequencing chromatogram. And the reason I'm just showing you this Sanger sequencing is that actually Illumina is producing an output that's very similar. So this is a chromatogram. So essentially, it's just DNA migrating into a capillary. But the point is, this lets us get a read sequence. So we have DNA in the input DNA, and then we have just a sequence for that DNA. But we also have the underlying measurements, technical measurement that gave rise to that sequence. And here, so in Sanger sequencing was just 204, and the same in Illumina, fluorescent bases. And so, for instance, if we see red, it's going to be a T. If we see a lot of green, it's going to be an A, and then black is going to be G. And the thing is, in some cases, it's going to be clear which base we have at which position. But in other cases, it is going to be a bit less clear which base is actually in a specific place. Like we see things that are a bit messy in some cases. And so not all bases are going to be as reliable as all the others, right? So some bases, they're going to be very clearly A, like for instance, this one, and some bases like this A here, that could also be a G because there's a lot of black as well. So essentially, when we talk about a read, typically we talk about 2 separate things. So the sequence of bases that the molecule has, and then the qualities of the inference for this sequence of base. And that's going to be 1 confidence score per base. And so this was for Sanger, but Illumina data is exactly the same. And the principle of the sequencing behind Illumina is very similar to Sanger, just scaled to a much higher throughput. And so we're going to get this sort of data where we also have one sequence with one quality score for each of the bases. And so the other thing is that Illumina has a higher throughput than Sanger, much higher throughput. And the consequence of this is just that, and that's true for high throughput data in genomics in general nowadays, is that we're going to deal with very, very large data set. So for that study that we're using the data of, the full data set, just for the raw reads, before we start any sort of processing, the full data set is 50 gigabytes. And so we're not going to work with all of this. And then more generally, it poses issues about just storing all the files and just transferring all these files, right? So this is not even a very large data set. So it's fairly common to have data sets that are even on the order of 1 terabyte. And then you're getting this link from the sequencing core where you end up having to download 1 terabyte of data. And this is not always like, this is not technically obvious. So I just wanted to make the point that we're not going to deal too much with this during the workshop because everything is kind of, I've been trying to reduce the size of the data as much as we could to make things feasible. But in a normal experiment, dealing with just the big files and dealing with big transfers and dealing with the long computation times are a real complexity. Yeah. So just to come back to these read files. So essentially, this is what I really meant, and what this means in practice by is meant in practice by a sequencing read. So this is an Illumina read, and this is just plain text. So it's actually a text file like this. So this box here, that would be the raw output of an Illumina sequencer. So that's the end product of a sequencing run, and all the technology behind Illumina is just this one text file. And then in this text file, we are going to have groups of four lines. Right? So we have, and that's going to be one read. And so we have a read name with some extra information. This here in bold is just a read name. So that corresponds to exactly where the read was. This coordinates here, they're where the read was on the chip, on the Illumina chip within. So I'm not sure if you've like ever looked in detail at the Illumina process, but like you just get a picture and then there's a spot at some point, and this coordinates. But it's just an identifier for the read, really. It doesn't matter more than this, it's just random. And then you have the read sequence, and then there's just like the plus line, it's just a spacer line, doesn't serve any purpose. And then there's a quality line. So we have, so the three parts really are the identifier of the read, the sequence of the read, and then the qualities for each of the bases of the read. Okay. And one thing we can right away notice is, the read ID is just an ID that's been arbitrarily given. The sequence, it's self-explanatory. It's just the sequence of the read. And then the one thing that's less obvious in FASTQ files is this quality line. And this, it's just, yeah, so like it's 9 lesser than AF. And everything. It's not just meaningful by itself. So I'm going to spend like just a few minutes explaining what this actually refers to, and really the fact that it's just ends up being just a quality. Each of these symbols is a quality between so 0 and 40 or 45. And essentially this is using the Fred scale, the so-called Fred scale. And it is a logarithmic scale where we're just writing the confidence as integers between 0 and 40 here. And so these are the formulas that we can use to go from the Phred score to the error rates or to the confidence. And so, for instance, a Phred score of 10 is going to be an error rate of 10 to the minus 1. And so, 0.1, and that's a confidence of 1 minus this. So that's a confidence of 90%. And then a Phred score of 20 is going to be 10 to the minus 2, confidence of 99%, and then etc. Phred score of 40, 10 to the minus 4. So this is like this. This is really just like the higher the Phred score, the higher the confidence, and it's logarithmic. And so the second thing is these are not integers between 0 and 40. So this is really the meaning of these symbols, but they're not directly integers between 0 and 40. Instead, the way this is done is that this all uses the Ascii encoding table. And so this is the Ascii codes. And this is a very old table. It's like dates back to like the beginning of computers. And so essentially, this is a table from 0 to 127. And it's actually used 7 bytes, not 7 bits, not 8. But the 8th bit was used for other things in the beginning. And so what this table gives us is just a correspondence between a byte number, so a number between 0 and 255, or 0 and 127, and then a letter or another meaning. So for instance, 72 is H, and then 101 is E. And so really what this means is that if we just like write several bytes in our computer memory, that can directly have a meaning in terms of a word, like in terms of characters or letters, right? So if we have this byte sequence somewhere in the memory of the computer, if we have the byte sequence 72, 101, 108, 108, and I can't see, anyway, whichever it is behind it, that just means hello. And whenever you see hello in a computer, or whenever you really see text on a computer, it's actually the ASCII code that's behind it. It's I mean, the beginning it was, it had like a couple competitors, but nowadays it's just only the ASCII code and like things that have built on the ASCII code and are still respecting the ASCII code. So anytime you see text on a computer, anywhere on a computer, you know that it is this ASCII code that's behind. So there's this hello here. It's also like stored by PowerPoint, and then somehow the computer, as it shows it on the screen, also uses this ASCII table on the left. And that's how you have like you go from like computer bytes, computer memory to text. So it's just like to introduce like the ASCII table, just because it's like such a fundamental thing in computer, and it's like every computer knows this ASCII table. And so for the purpose of RNA-seq and genomics, what we're going to use is and this encoding of Phred scores. What we're going to use is the reverse property, where we're writing a letter and we're actually meaning a number. And so, for instance, if we write A, we can look up A in the ASCII table, and that's 65. And the next thing to notice is that if we look at the characters, like the numbers 33 to 126, these are actually text characters. Like the rest is not text characters. Like this is end of transmission. This is something that dates back from, I don't know, communication needs of early computers. They just mean literally end of transmission. So think of like Morse code or this sort of things, and the things you might need to just have to have two devices communicate together. And so all of these characters on the left, on the first column, and then this one, this is space. They're not really usable in text files just because they're not meaning anything obvious. And so what we're going to use for our encoding here for Fred scores is typically these 40 characters here, starting at 33. And that's, I'm not sure if you've ever heard the name Fred 33, which is the name of the major Fred encoding. It's just like the Fred score encoded starting at character 33. And so, yeah, so, and essentially the exclamation point is going to correspond to a Fred score of 0. And like this is going to be a Fred score of 10 with the plus, and then arriving all the way to Fred scores of like 30, 40 with capital letters here. Yeah, so this is, and then we have encoding from 0 to 40, like this. And this is really what the Fred scores correspond to. So essentially, if we go back to our FASTQ file, we have our quality scores line here. And each of these characters correspond to one number between 0 and 40. So for instance, this 9 here corresponds to the code 57. And then the greater, the less than sign correspond to code 60. And then we remove 33 because of the starting point of the table, in the table. And then meaning these characters here have correspond to Fred scores or confidence scores of 24, 27, 32, etc. And then we can also like go back to the actual confidence scores in terms of percentages, and that would be 99.6, 99.8, 99.9, etc. So really, these characters are each one number between 0 and 40, and the higher the number, the better the confidence for the base that corresponds. Okay, any questions on FASTQ files? Is this clear? This idea of like 4 lines per 4 lines per read, and then we have the read sequence, and then each of the bases have a specific quality or error rate. Yeah, and so I hope this looks simple. Like it's really just these FASTQ files are the basis of a lot of things in genomics. Pretty much everything uses them. Yeah, so if that's clear for everyone, or if we just want to like start working on these files directly, we're just going to switch to the exercises. Yeah, so today's exercises are going to be raw reads. I have put a PDF with the exercises in the Google Drive directory. I'm going to try to open that. Okay, this is what it looks like. So this is something that's new. So I don't think there are typos in it, but it might still be possible. I've not like, it's not been debugged entirely yet by any students. So there might be some errors, but I don't think so. This PDF should be in the Google Drive. So if you don't have it, and today we're going to do exercise 1, exercise 2, and if we have time, trying to go into exercise 3. So I'll let you just like open the PDF, set up your computer, and if you have any questions, the beginning is really just connecting to the cluster, and then just managing to connect to the cluster and getting started with the workshop's files. And that's probably a lot more difficult than it sounds, just because of, I mean, if you're not used with Unix, it's always the learning curve is what it is, and you just have to try and try again until things work. Share again the link. Yes. Oh, I had shared the link, but not to everyone. Okay, has everyone been able to access this this PDF? Okay, great. So it is in the folder. And then I think, I mean, the way this works best, and of course, I mean, it's always up to you, would be if we really just work together. You're all going to have issues with the Unix commands, and really this works best if we can see each other's terminal screens. So I mean, we can't share at the same time, but if you have an issue, please just like be ready to share your terminal, and then like it's normal if you have errors. That's kind of why we're here. So yeah, the goal is to really try to work collaboratively, and usually I mean, when we're doing the workshop in person in the QC bio space, people work by small groups of 2 or 3, and of course we can't do this here. But if you want to, says permission denied. Oh, that's possible. Thought I had tried to. Okay, that's weird. Oh, I see. Okay, it should be good. It should be good now. Yeah, that's I had like forgotten one character in my command, and that meant that nothing worked. So and I've been like doing Unix commands for years and years. So yeah, I mean, errors when you're doing Unix, it's just really, you mustn't be afraid of trial and error. You know, it's just the way it works for programming in general. And then you learn one thing at a time, and then And then yeah, just get comfortable with one more thing. And this is I mentioned the full data set is 50 gigabytes, and we're just going to work with like a small subset of the reads for 2 samples out of like a couple dozens. And that's, I think, 200 megabytes. And I really expect the Unix levels to be quite spread out in this class, and that's fine. I mean, if you're going faster, then I mean, good for you, and you can like go through all the exercises if that works. And if you're slower, then that's great, and we can look at your code, and I can discuss the solutions, guide you through the solutions and everything. And in general as well, the yeah, discussing the solution is going to be good for everyone. So all questions are and all problems are really welcome. Has anyone connected to the cluster? Has anyone not connected to the cluster, or has everyone connected to the cluster? Okay, so is it an issue with your password, or to use? Yeah, what's the issue? Or what's the error message? Is it just an issue with your password? That wasn't my intention. I'm not sure. I mean, if you've like learned that one, this might be an option. But I've never done it. Are you using Mac or Windows? Okay. Do you have the Ubuntu app? I'm not sure. I'm not so. I sent out an email, but it was like a week ago. Depending on when you registered for the class, you might have or not gotten it. If not, okay. So on Windows, the simplest way, the best way to just really gain access to a terminal, the proper Unix terminal, is to install the Ubuntu app from the Windows Store. And Ubuntu is one of the main Linux distributions. Yeah, some people prefer PuTTY. Both work. You may be installing PuTTY is pretty much faster than installing the Ubuntu app. So if you just want to catch up with a class, just install PuTTY maybe. And then I can try to find online a tutorial to install PuTTY. That's not directly. Yes, I have your email probably in the table. Does anyone else need the email? Okay, I'm going to just write down that you should have the email. I'm going to write down that. Maya, which question is this for? Yeah, so the first exercise 1, I believe, is really just to get started with Unix. Just some small obtaining the workshop data. So we're talking about exercise 1C, right?
Yes.
Yeah. So I mean, you can use cat as part of the solution. Cat itself is not. I mean, you can use cat and just count the samples by hand. So this is a question about the number of samples, right? 1C question 1.
Yeah, but in the other ones, you also need to look through the tables, and it doesn't seem efficient.
Yeah, no, that's, I mean, and in Unix, a lot of times you're just going to have like a million different ways of doing the same thing. And I mean, counting by hand is possible. And sometimes it's just like the most efficient way, honestly, like doing things by hand. Yeah, I'm just going to, you can use, so, okay, one command that's also very good to know is the WC command. Yep. What WC stands for word count. And so that's actually going to, and the L -L. So -L is to give an option. And that stands for line, the L in line. And otherwise, by default, the command outputs the number of characters or the number of bytes in the file. And as we have seen, like the 1 character corresponds to 1 byte when you're dealing with like those basic Latin characters. And then, yeah, so number of bytes, number of words, where it takes its name from. So that's just like white space separated. So either spaces or tabs or new lines. And then just the number of lines. And just like counting the number of lines is a very common thing in bioinformatics. So it's really useful to know that one. And Gloria, did you go for Ubuntu or PuTTY?
I'm trying PuTTY.
Okay, so you're going to, so this SSH command, if you're using PuTTY, you're not doing SSH. Instead, you're just going to have buttons on which I don't know exactly how I don't remember exactly how it's asking you things. But you're somewhere you're going to have to enter the address of the Hoffman 2 cluster, which is the address after the @ in the SSH command line from
Okay,
from
yeah,
the
I see.
And then you're going to have to enter your username, and then you're going to have to enter your password in yet another field. And if it asks you for a port, it's going to be 22, but that's pretty the default already in there. Oh, they have a good Windows. Yeah, the good thing about installing the Ubuntu and the reason why I recommend it on Windows, the good thing about installing the Windows subsystem for Linux is that you really just get terminal and then you can do SSH as you can find it.
Okay, yeah, I can try that
online. One. It's just, I'm worried it's going to take an hour or so to install and download and everything. But it's just there from the Windows store, so you don't have to do anything special. And then you just click on it and get the terminal, and that's
okay, that sounds more straightforward.
Okay, so I'm just going to go through the first exercise. Oh, I need the section first. And if there is anything not clear about the PDF, just let me know, because that way I can update it. Okay, so I'm just going to show for the exercise, the first exercise, which is really exercise 1C. So we have these directories here. And then the first question is counting the number of samples in samples.txt. And so as Mohammed said, the most direct way of asking this question is literally to do wc-l, which is a command that directly asks how many lines are there in this file. One thing I really can encourage you to do is to use the less text viewer, to learn how to use the less text viewer. And I'm not sure how many of you have tried with opening the file with less. Does any of you have tried opening the file with less? No one? Okay, so as far as I'm concerned, that whenever I have a text file, like opening it with less is almost always the first thing I'm going to do with it, like almost always. And so less, it's called less, and the reason for the name is that it comes after a program that's called more, and I don't know where the name for the more program comes from. But anyway, this is the normal program you would use to see a text file. And so you use the less command, and then the name of the file you want to look at. And you get this screen like this. And you can move up and down with the arrows, although I mean, at the moment, I can't move anywhere just because the entire file is already on screen. And it just lets me see the file like this. And once you're into less, like you can't, I mean, very often you'll feel like you're stuck. And so if you're stuck, you have to know that you have shortcuts to navigate inside less. And especially to quit the program is the Q key. Just hit the Q key and that's going to quit the program, like go back to the terminal. Okay, and so it's interactive. And so where it comes in handy is first you can just confirm that you actually have one sample name per line, which is just confirming that you're actually, when you're counting line, you're actually counting the samples, and then you're just like able to see anything going on with the file. And so here, so we have 30 samples. Second question was about the table. Why did I not? Oh, the PDF over there. And the second question is about the average read length. And so again, and that's from this other file. Oh yeah, and here I'm using copy paste. Copy paste is really good in any bioinformatic or coding related questions. With I mean, at least it's way better than entering things by hand, like writing things by hand. At some point, it's copy paste I mean, becomes still a human manipulation and still has issues, but it is, I mean, it is a good thing to copy paste as much as you can instead of copying things by hand. One, because it saves time, and two, because you don't introduce typos. The second way to not introduce typos is to use tab completion, so auto completion. And so here I can just write less and then Kim, and then I want to complete the 2016 info table.tsv name. I can just hit tab and it's going to complete the name. And I can, and for instance, if I had made a typo in what I had already typed, and let's say I replaced the 6 by the 5, and then I'm trying to autocomplete, I'm hitting tab, it doesn't do anything because there's a typo, so there's no matching file. So what this means is that anytime it autocompletes, it means that it has, like the computer has confirmed that there was no typos. And that's the main reason why you autocomplete, is because it ensures that you don't have typos, or at least you notice the typos much earlier, right? It doesn't autocomplete, you check for typos right away, and you know you don't have an error anymore. Anyway, so yeah, so we can open the file like this, and it doesn't work as well as before. And the reason this is a header line here. And so it's one line. It's just the header line for the table, but it wraps around and continues a second line. And when this happens, what we can do is actually use less with the capital S option. And now we have the header line that just keeps going on the right, and we can move with the arrow keys to the rest of the table. But still, like, all the fields are shifted. The columns are not displaying properly. And so what we can do is also with less, and then we can use the S option, and we can use longer tabs to just make the columns wider. And this lets us just look at this file in a bit more cleaner way. And here, actually, there is still like an issue here. I'm missing one character. And then here as well. So I'm just going to increase it a little bit more. And now the columns are not shifted anymore. And so we have this average read length column that corresponds to our sample. And so the average read length is 76 in all samples. And I just realized that it's actually 85 for this one, which I hadn't noticed before. I wonder if this is just a typo. 676. I really wonder if it's not just a typo of the person who has entered the data in this table. So this is the data that was uploaded to the NCBI for archival. The other option, if you don't want to use less, so this is one way you can really manipulate this table and kind of browse this table. The other option is just to download the table to your laptop, open it with Microsoft Excel. TSV files, they're just like CSV file. They're very normal format to work on with any Excel sheet program. So the third question was about this genotype. So here we have the genotype column, and then some of the samples that have this GFP construct, which is used for sorting the cells, so making the tissue more pure to reduce the noise in the experiment. And they all have this construct, right? They're just this modified line that they've been working with to do the cell sorting. And then some of these samples have this NRL --, so the knockout of this one transcription factor that the authors were interested in. And so to count this, we can just, as Mohammed said, we can use grep. And this is just matching all the lines from our file that have this NRL chain of characters in it. So if we again, oh yeah, and so for less, anytime you want the output to be more readable. So here, when I do grep, it just outputs on the terminal. Instead, we can use a pipe character to send it into another command, and in this case, that's going to be less. So this one command here, so it's a compound command with two commands in it, with a pipe in the middle, and so it takes the output of the first command and gives it to the second command, and then the second command executes. So here, this is less, and this is our output from the first from grep. And again, we can just put it on the single line. And now all of our lines, they're all the lines that have the NRL minus minus. And again, we can do just counting the lines. These are 14 lines, so that would be 14 out of 30 samples. We can also, there's like 50 ways to do it. We can also just count the number of matches of matching lines, which is also 14. One way that's very useful to know is actually doing a table of what's in a specific column of a file. So here, so what we're interested in is the genotype column. So that's the 5th column. And to extract the 5th column, we can use the cut command. And then cut and the 5th field. The F is for field, and that's for column. And then and we want column number 5. And that way we can just extract the 5th column from the table, from the TSV table. So TSV tables are really common in Unix. And then we can use a command combination that's very, very common, and which is sort and unique together. So we take this column here, which we generate with the cut command, and then we send this column into sort, to just sort the column, and then into unique with this -c option to count the number of occurrences of each of the lines. And what this gives us is it just counts the number of occurrences of each line in this column. And so these are the number of times, these are the content of the 5th column. And so we have the header line, and then we have 16 NRLP GFP mice, and then 14 NRLP GFP NRL minus minus mice. So here we have 16 and 14 samples from the two groups. Okay, so I just wanted to show you this like sort unique command combination that lets you do contingency tables in Unix. Very practical one. Any questions? Okay, any questions on the first exercise, or is anyone moving toward the second one? One more thing I can show you, and one command that's also extremely useful and really have seen it before, is the head command. And so you just type head samples.txt and you get the first 10 lines of your file. So it is an alternative to head. Some people by default do head on their text files. I prefer doing less, just because it gives me more options. But so yeah, so here, yeah, this is the entire file. And then when we do head on it, we get only the first 10 lines. And then we can choose how many lines we want. So for instance, if I just want the first 8 lines, then I do head -n8. Okay, if I just want the first 4 lines, then at -N4. And so this kind of brings us to the beginning of the second exercise. And we're going to be working with this P10KO rep1 FASTQ file. And so this is for the P10KO rep1 sample. And again, this sample, it's like the P10 developmental stage. And then this is one of the knockout samples. And then there's the first replicate for this particular combination of developmental stage and genotype. And so we're working with this file. And then, okay, so the first thing we can do is doing a head of the file. This is the most, okay, and this is what it looks like. And anytime you see this is like, and the actual way it looks might depend on your terminal. But essentially, when you start seeing these like weird characters here, so question marks in my case in the diamond, it means you're looking at a binary file in text mode. You're trying to look at the binary file as if it was text. And this is binary because it's compressed. So any compressed file is always going to be binary. And so what we need to do is to decompress the file with, for instance, zcat. And we can do like this. So we decompress the file, and then we head. So head is going to see the decompressed file. And then we can take the first 4 lines, which, because this is a FASTQ file, is going to be the first read. And so we have the ID of the read, and then the sequence, and then the qualities. But you really have to decompress the file because it's gzip. The other way you can do things is just unzip the file entirely. But it's not very good practice to just unzip the files entirely. It's better to do it on the fly within a pipe construct, just because unzipped files are just going to take huge amount of space on your disk, and disk space is maybe the biggest issue in terms of managing a big bioinformatic project. So generally, you don't want to unzip your files just because it's going to create even more problems. Although you can always like zip and unzip the files back and forth, and it's like no data is lost. It's not like no data is lost. It's like a lossless compression. Essentially, the key idea behind this sort of compressions is that, so we have this sequence of A's here. And so instead of writing, I don't know, like, is this like 15 A's? It's maybe going to write something like 15A like this, right? And this is the essence of these compression methods, where you can notice things that occur multiple times and just write the same information in a more compact way. So keep your file gzipped, and then you can zcat them, or equivalently, so you have zcat. And then the other command you can use, which is equivalent. Like zcat is exactly the same as using gzip, and then cd stands for, it stands for this. So this gzip cd and gzip decompress standard out are the same thing. And standard out is just the terminal or the pipe. So in particular, if I can do this, and this is the same as doing zcat, right? One thing you have to know is zcat on a Mac. So I mean, here, even those of you who are using Mac, and I'm using a Mac, zcat here is the zcat from the cluster. So that's I mean, sort of the normal zcat. On a Mac terminal, if you're just working on your own Mac, zcat is going to be different. You can't use zcat for gzip files. So I just use gzip cd all the time, just because I don't have to think where I am, and if I'm on my Mac or if I'm on the cluster. But they're the same. Like the zcat and the gzip cd or the gzip decompress to standard out, they're all exactly the same thing. Yeah. So this is how you handle zip file, gzip file, or oh, I don't have my video anymore. Does anyone have an answer for the first question? So in exercise 2A, item 1. How have you been able to measure the read length? Anyone? So what have you tried? Does anyone have an idea of how to do this?
I'm assuming there's some way with word count to specify a row, but I haven't figured it out.
Oh, yeah. So getting one specific row is so okay. So one way you can do it, honestly, is just like, so if you do with head and you show the sequence as I just did, you can copy paste the sequence and then just repeat the sequence with echo. So that's one way. I'm going to show how to extract a specific line as well.
So you mean just like highlighting the row, copying it back into the command line with echo?
Yep.
Okay.
Yeah, I mean, as long as the task is so generally the main issue we're worried about with programming and Unix and everything is not introducing typos or not introducing human error. But here, just if it's just copy pasting the entire sequence of one read, that's not going to be a problem. Oh, there's this.
So, but echo just spits the text back. It doesn't actually give you like a numerical result.
Yeah, yeah, but you can combine it with WC.
I see what you mean.
Yeah, just like it's just like trying to. I mean, so many. I mean, most of the time, if you're talking about Unix commands, I'm not talking about like bioinformatic programs and I mean, even then you have a choice between different programs to do the same task, just because this is the way academia works. But if you're talking about Unix tasks, you normally have several ways to do it, and any of them is good. Okay, so I'll show a couple ways. And so the way I just mentioned was just selecting that line And so copy pasting it to the terminal. By the way, some commands that are really useful, some not commands, some shortcuts that are really useful in the terminal. And so the first of them is, you've seen this like #C characters in a lot of my commands here. That appears whenever I press Control-C. So whenever I press Control-C, I get this like #C. I'm not actually entering #C. It's like the # stands for control, like the control key on your keyboard, and that kind of stops the current command, right? So I can, if I start typing a command and I'm just like, oh wait, I actually needed to do something else first, then I can just like go back to the beginning. I don't have to erase the entire command before I can start a new one. The other one is the other two ones that are very useful in their paired series is you can do Control-A as in alpha and Control-E as in echo to go back to the beginning of and the end of the line. So you have Control-C, Control-A, and Control-E. They're just very useful shortcuts to just navigate your command line. And so I can echo this, and what this is going to do is just echo this entire line here. And so, oh yeah, and then the arrows. Up arrow gives you the previous command like this, or you can navigate your command history with up arrow, and then of course tab, which I already mentioned before. And so we do this echo and then WC. And so we get this. We get one line, one word, because we don't have any white space, and then 77 characters. And does this match the number we had in the table?
No.
So we had 76. Actually, the reason is there's a new line there. When echo prints this argument that we gave it, it adds a new line at the end. So going from there to there and back at the beginning of the line. And what we can do to avoid this is add the -n option, which is for no new lines or something like this. And then we're going to get something like that. And then we don't have this new line then. There's nothing there. And so now if we just use echo with a -n and word count, then now we have this 76, which matches what we had in the table. And there are plenty of ways to do it. One way, so and then what if we don't want to use echo and copy paste? What if we don't want to copy paste and just like ask for the length of the second line in the file? No, not like this. So again, we can go back to our zcatting of the file and use autocomplete. And then we can open show the first 4 lines. And then if we can open the first 4, we can actually open the first 2 as well, right? And you just get, and then the sequence is the last line. And similarly to head, we actually have another command to extract just the last lines of the file. And the same way we can choose how many lines we want with head, we can choose how many lines we want with tail. And so if we just pick the last line of the first 2 lines, then we actually get the second line. So that's one very normal way to extract the nth line of the file, is to use head and tail like this. And then we can do word count, counting the characters. And again, we have this 77, which has the same issues as before, is because each of our lines in the FASTQ file ends with a new line character. One thing we can do also to remove this new line is using the tr command, which is the I think transliterate. Anyways, tr command, And we can use it like this. So we can ask, so generally the tr command lets you convert one character to another character. So for instance, I could convert all the capital A's to lowercase a's. And now all my A's are lowercase. So this is the basic use of the tr command, but we can also use it to delete specific characters. And in this case, we can delete the new line character, which we can write like this with \n, and that means the new line character. And again, this same thing that happened with echo when we added the no new line option is happening, where we're printing the line, but then we're not adding a new line at the end of the line. So the next command starts on the same line because the new line is missing. And so now that we're sure that we don't have our new line, we can do word count and get this 76. Okay, does this make sense for everyone?
This does make a lot of sense. I'm able to follow along. I'm wondering if you're going to add what we do into the PDF file at the end of the course.
Yes, I'm going to provide solutions for the exercises, like in the same form as for the separate PDF, but.
Thank you.
So one thing I can show about FASTQ file is kind of a not very well-known tip, but it's often very helpful to like build these small questions. And I'm not going to really explain how it works, but essentially, so we can show the first 8 lines here. And we get the first 4 lines are the first read. The next 4 lines are the second read, right? And so we have these groups of 4 lines. And there's kind of this magic Unix command, which is paste, and then with 4 hyphens. And if you're interested in knowing what the 4 hyphens do, you can sort of find them and try to find the explanation on the internet, but it's kind of an advanced thing. But the fact is, when you're working with FASTQ file, it's just kind of magical. It does like this. So this is opening in less the output. Oh, let's not use less. It just regroups line by groups of 4. So it takes the first 4 lines, put them on one line with tabs in the middle. This is the same thing for the second line. And so, for instance, I can then take the second column, and that's going to be just the sequences. Anyway, this is something that's useful for FASTQ files. FASTQ files are kind of a remnant of like the early days of bioinformatics. They're not a very efficient format. They're really weird to work with, and there is like Fred score encoding that's a bit interesting. But it is what it is. We just inherited this from 20 years ago, and it is still the normal format for all Illumina data. Yeah, so now that we have these like files, we can like, I don't know, try to look at the statistics of how long these reads are over the entire file, or just how many reads we have. Or I'm going to show you this in a second. Okay, any attempts for the next exercise? So the first base call of the first read, what's the confidence for it? And again, so the first read, that's just the first 4 lines. And so what's the Fred score? What's the Fred character? What does the score character for the read. So for the first base, so that would be this G here. And so the quality that comes with the G, which character is it, and then which score does that correspond to? Is this?
Yeah, so like right below it, coincidentally, it's also a G. Looks like.
So this is also G. And then the G, to understand what the G actually means, you actually have to go back to the Ascii table. So you can go back to the PowerPoint. And so like this. Having trouble with. So we can go back to the PowerPoint, and then G is here, so it's 71. And then we can do 71 minus 33, which is 38. And then 38 is going to be something like, close to 99.99%. At least something higher than, much higher than 99.9%. And we'd have to just, open a calculator of some sort to get the actual confidence score. But it is a Phred score of 38. And generally for Phred scores, so anything under 20 is not very good. And then for Illumina, the normal scores are around 30, 35, up to 40. 40 is the thing is, if you could have score way above 40, but there's always a risk that other sources of noise that you're not accounting for actually making you underestimate the error. So yeah, pushing the score too high doesn't really mean anything. Yeah, so that was for the second question. And then how many lines does the file have in total? So that's actually going to be the same, the exact same command as we use for this. Not exact same, but the very similar command we use for the samples.txt file, just counting the number of samples. And the same way we counted the number of samples, we can count the number of lines in our FASTQ file. And here we do have to be careful because this file again is compressed. And so the characters in this compressed file are essentially random. And among those random characters, we have new line characters. So if you just try the WC command on the compressed file, it's going to work. It's going to output something. But all it does actually is counting the number of randomly occurring bytes that correspond to a new line character for just at random. It's not actually counting the lines because you don't have lines in a binary file, in a non-text file. So we can do it like this. I think it takes like a few seconds. Yeah. And so here we have this many lines. How many samples, how many reads does this correspond to?
So like a fourth of that?
Yeah, so we take this number divided by 4. There are many ways to do this. One way is with R. I'm just open R, copy paste this number divided by 4. And that gives us 2.2 million reads. Yeah, that's just anyway. Open a calculator and open it. Another way we can do it is actually so this is just the counting of lines. But as I mentioned before, we have this sort of magic command for FASTQ files that lets us just put 1 reads per line, where we have so read 1, read 2, read 3, read 4, etc. And what this means is that if we do this with if we count the lines now, we're actually counting the number of reads right away. So Unix just likes to work with lines and columns and tab delimited columns. Like everything in Unix is made to work with lines and columns. I mean, not everything, but a lot of tools or Unix tools are made for that. So when you can have your data in that format, then everything becomes a lot easier to do. That said, yet the easiest way to just the easiest way to just look at your FASTQ files just to ask a bioinformatic tool to do things for you, and that's what we're going to do now. Is using FastQC to just ask a number of questions about our FASTQ file. One thing I can mention though is this command here that's at the end of the screen. That's typically something that I use when I get data back. So just like I get data back for a number of samples. That's just a very quick way of confirming how many reads I have exactly for each of my samples. Which is the basic information you want. That's like the most basic information you want to start with. Oh, and by the way, so this number here, 2 million reads, that's very small for mammalian RNA-seq experiment. Normally, a mammalian RNA-seq experiment, it's from anywhere from 10 million to 40-ish million, to 10 to 40 million reads per sample. And the reason we have like this like depressed number here is because we're just using a small subset of the dataset. Just because, I mean, so today, like the commands are fairly fast to run. It still takes 5 seconds here to just count the lines. If we had the full dataset, which is 20 times bigger, it would take a minute or two. So we'd just sit there, wait for a minute or two, and then for the more computationally intensive steps we're going to do tomorrow, as you're going to see, everything takes actually more time, even with a small fraction of the dataset. Yeah, so computation times are a factor when you're working with Unix. It's just like hits your productivity just because you're just waiting for things to complete. Maybe you're just waiting and then there's an error and you have to start again. But that's just the way bioinformatics are. Is you just have to do this sort of time management where yeah, you're trying to do things and they don't always work, or you can't necessarily do the next thing right away, and then you're trying again and wait for some time. It's really like waiting for incubations or something. That's the way I see it. So you have to plan accordingly, and this is sometimes hard. It's not like a molecular protocol where you know exactly how long this and that are going to be. It's more, yeah, there's just like trying your best doing time management.
So I think I missed the magic. I know you didn't really explain exactly what it does, but the last output that we got, is that for the last read? There's 4 reads, and I'm assuming they're not all the same length.
Which output? Are we talking about this command here? Or
yes, that one.
So this is counting the lines in the entire file. So I'm not using head here. So I'm just getting the entire file. Like I could hopefully that's not going to. Yeah, this is just like the 2 million reads that are just like yeah, it's kind of stuck now. Well, again, oh yeah, another thing is when you have like this, for some reason, I mean, it wasn't going to finish. But yeah, you can use Control-C to interrupt a command that's running and you want to just interrupt. So this is just like taking all the lines, all the reads with one read per line here, where we have like the ID, the sequence, and then the qualities. And just counting how many lines we have. And that gives me the number of reads. If we go back to, yeah, that one. This was the command we used to count the number of bases in the first read, right? And so again, so if we like really, when we have these commands with like pipes like this, like it's really series of pipes, and that's where I think that's where the word pipelines come from. But maybe not. Maybe they just reuse the pipe because the word pipeline already existed. And I'm not going to, and essentially you can break down the first steps. And I'm just going to start like this. I'm not going to show just this zcat command because it's just going to show the entire PySQ file again on the screen. But we can start like this head-end2 command. It does this, it extracts this first two lines, and then we can add the next piece of the command. And that's like out of those two lines, extracting the last line. So that's the second line. And now we're left with just the last line, which is just the sequence line. And then we can add the TR deletion of the new line character at the end. So like this new line, I'm talking about this new line character here, like which the one that's selected now, that goes from this A and then back at the beginning of the line into the bracket. That's a new line character. And now if I'm removing that character, what's going to happen is that there's this one thing that was here between the A and the new line. It's not there anymore. The character is not there. So there. And then I can count the number of, bytes in that sequence here. That's for the first read. So if I wanted to look at the sequence of the second read, so here so these are the first 8 lines, right? So and this first 4 lines are the first read. The second line is the first read. And if I wanted the sequence of the second read, I would take 4 plus 2. And so that's going to be the fixed line. So here, if I'm doing head and 6, then the last line is my second sequence. And then I can just extract the last line again. So before I was doing head and 2, tail and 1 to get the second line. And then now to get the 6th line, I get head and 6, tail and 1. And then I get the second sequence. And I can apply the same. It is, but I mean, we can see it's the same length, but I could apply the same series of and again, you have many ways to do this. I want to make things more confusing. So I'm just going to show this one, which is maybe the most like basic one. And we could do this for all of the sequences. And if we wanted to do like a histogram of all the sequences, we could, I mean, do other like more complex command. We're not very far away from that. But just, yeah, I mean, we could do it as well. It's just a bit more complicated, slightly more complicated. Yeah, the big difference between this command and the commands to count the total number of reads is that here we're using this head command to truncate the file. And right away, like this file has 10-ish, how many was it? It was 9 million lines. And then here we're just like taking the first 6 and truncating the file. So it's a very big difference compared to working with the entire 9 million lines. Yeah, it's this is why we're practicing with these files. But now we're just going to switch to like a bioinformatics software, and a lot of problems in bioinformatics. I think it's very important to understand what information is in the files, like understand the file formats, so that we know what information we're actually dealing with, right? So here, FASTQ file is just X reads or N reads with some sequences with some length, and then associated base qualities. But for most of the operations, we're actually going to have bioinformatics software do it for us. So in this case, FastQ. Has anyone finished running FastQC? And the QC in FastQC is kind of a play in words. It's like, so it's FastQ and the same time QC, which is quality control. And so the first thing is run FastQC, and then the second thing is download the HTML report, and that's going to show you a bunch of graphics. And in principle, that's when we should be starting to get our first path problems. It's like file not found and this sort of things, especially for the FastQ program. Although, did I put the full path in the document? Yeah, I put the full path there, so it should be fine. Okay, could someone? So this one is easier. Could someone just show what they got for the FastQC output on the command line? You know, I wonder if how long has it been running for?
Mine was only less than a minute.
Okay, yeah. So can you just show so that everybody has an idea of? Can anyone?
You disabled screen share.
Oh yeah, okay, it's enabled. Yeah, it should be enabled. So did anyone have problems with running it? Yeah. So this is so the program here just like informs us on like how far it is into the file. And again, this is very fast, but as I mentioned, sometimes some of these programs are going to run for several hours, and you're pretty happy to know whether you are at 90% or if you are at 20% after 2 hours. Because if you're at 20% after 2 hours, then it's pretty not going to have your results. COVID. Anyway. Yeah, and so this is sort of what the output looks like. So you have these sections. So on the left, you have this like kind of outline of the different sections. So you have statistics, per base qualities, per sequence qualities, and they're all like either box plots or histograms. And the first thing is just like confirms which file we're talking about. By the Sanger Illumina 1.9 encoding, they actually mean FRED33. It's just at some point, like in the early 2010s, for some reason, so the Sanger tradition had been to use FRED33. So starting at character 33 in the Ascii table. And then the early Illumina machines started using 64, I think, or they started changing the offset. And then they realized that it was a dumb thing to do, and just like it was better if ever you were using the same one. And so they went back to thread 33, and now you don't really, except for very old data, you don't really have to think about other issues. It's always thread 33, but that's why it's called like Sanger/Lumina 1.9. And then the total number of sequences should match what we had at the terminal. Sequence lengths, and then it also gives you the overall GC%. And so the first histogram it gives you is like what are the scores? What are the thread scores? So between 0 and 40, what are the thread scores from the first base to the last base of the read? So from base 1 to base 76, what are the quality scores like? And what we can see that there's like a slight, and this is a good run, especially modern Illumina machines, modern Illumina instruments. They've become very reliable. I mean, much more than 10 years ago, when people built the knowledge on I'm not sure how old FastQC is, but a lot of these tools they were built like around 10 years ago, or I mean, depending. And so we do see still a drop in quality from base 1 to base 7, from the beginning to the end of the read. And these are like moderately short reads. And this is not even like the latest generation of Illumina machine. But essentially, even at the end of reads, like the typical quality is way above 20. So as I mentioned, Illumina, like between 20 and 30, the quality is like so. Under 20 is really subpar, and then above 30 is really good. Like that's 1 in 1,000 chance for an error, 10 to the -3. So yeah, essentially, like the quality overall is good. Although at the very end of reads, we do see this drop of quality because Illumina is a sequence by sequencing. Sequencing by synthesis technology. In contrast with, oh well, I guess anyway. So it is a sequencing sequencing by synthesis technologies like Sanger. So we have the same phenomenon where the further into the read you are, the worse the quality gets. Yeah, the second one is just asking whether we have a subset of reads, and this is like a good result. It's just asking whether there is a subset of reads that have a lower quality, and sometimes you see a bump in the middle, which may or may not be an issue. Sometimes just an issue is specific tiles of the flow cell, and in that case it's not a very big issue. Sometimes it can reveal an issue with a sample. And generally here, and the reason why the exercise PDF asks you to compare the two samples together is we're not sure exactly what we're looking for in terms of what the data should or should not be like. We have some general ideas about like, okay, the qualities should be above 30 for most of the read. And then the reads should be homogeneous. And a few others I'm going to mention in a minute. But the main criterion I would say is really, if we're looking for outlier samples, like maybe RNA extractions that haven't worked properly or issues with library prep, all samples should be similarly good or similarly bad. And if there are big differences between our samples, that probably suggests that something went wrong in somewhere in the experiment. So maybe one sample is very different from all the others for no reason. That suggests that this is a technical outlier. And sometimes it's not very clear. I mean, FastQ, just looking at the FastQ files there, it's not a very powerful way to detect that this or that artifact has happened. But it kind of, sometimes we start seeing things, very often even, we start seeing things in the FastQC report where we're just like, this sample might be a bit different from the others. And then the further we progress into the analysis, and then the more we realize that, yeah, this sample was just something was wrong. And the fact that this sample was already looking wrong at the read level, so at the raw data level, that really suggests that there is a technical artifact. And also, if there are really big technical artifacts, we can see them right away in the, like if there's for some reason the sequencing didn't work, and pray that's not going to happen with modern Illumina sequencing, then we see it right away. Okay, so this one is interesting. The per base sequence content. This one is so the next two ones actually they're paired. So there is pair base sequence content and the overall GC content right after that. And so essentially what we see here, and that's what we're going to see for the Gaussian. So that's a GC content, and so this one in this case it's not. I mean it's good. And then if we go back to the previous one, yeah. So that's typical for RNA-seq experiments. So for the first 10 bases or so, we're seeing biased composition, and that's because of the reverse complementation priming. It's just like it's supposed to be random. It's not perfectly random. So we're just seeing these biases in RNA-seq. And so FastQC is telling us that this is biased. This is like the pair base. There's like this red cross near the per base sequence content. But this is actually normal for specifically RNA-seq data, just because those first bases, because of the priming of the reverse complementation in the library preparation step. So at the molecular, at the bench, that's biased. So we actually expect those 10-ish bases to be biased. And then it's kind of stabilizes. And what we're after, especially, and then that's typical for mammals. But in some species, sometimes like some species have a GC content that's very biased toward either IT or GC, and that then the A and T or the G and C can be different. But what we don't expect is that the A and the T would like stabilize at different levels, or that the G and the C would stabilize at different levels. In general, what this suggests, and that's also something normally the next graph also shows, is if we have peaks at weird places in the next graph, that can be due to ribosomal RNA contamination. Like if we see big peaks here, because ribosomal RNA is like single-stranded and it's always the same sequence and it's like so abundant. Because it's single-stranded, we don't have this property that the A and T should be more or less the same. And genes are also single-stranded, but like overall they average out. Whereas ribosomal RNA as a single highly abundant species, they tend to create problems. So if this graph here is really bad, it's like sometimes you see like multiple peaks and very weird spiky things. That can be an indication that there is ribosomal RNA contamination. I can, yeah, and the next one, yeah, this is like ends. So sometimes Illumina sequencer just, instead of writing A, T, G, C, G, T in a read, is just going to write an N. So if usually then they're not very abundant. If they're very abundant, something went wrong with the sequencing. Sequence length distribution, the next one, that's like all the sequences are 76, so that's also normal when it just comes out of the sequencer. When we're going to clean the reads afterwards in the next exercise, so we're going to clean the reads, and that is going to introduce like differences in sequence length. But like, I mean, generally you paid for, I mean, you ask the sequencing core for 75, 76 bases of sequencing, and then that's what the core did. So that should correspond to just what do you ask the core. Duplication, often, okay, this one is not actually a, so these are the sequences that are seen many times. And usually for RNA-seq, we're going to see a peak at like at 10 or more, or 50 or more, or 100 or more. So we're actually going to see an excess of sequences that are present many times. And that's just because in a transcriptome, some genes are more highly expressed than others. Like let's say like actin is going to have a lot of reads on it. So the sequences from actin are going to be present many times, or I mean, this sort of things. Yeah, and I think that's the other one. Oh yeah, adapter content. I was just showing adapter contamination, and that's fine. So that's also an issue with library preparation, and I've rarely seen it really, really bad. Usually it's detected before the library sequence. So normally there is like quality checks of the library before it's put into the sequencer. So no, normally we don't see this one as being bad. Okay, and the other sample essentially the same.
Can I have a question?
Yeah, of course.
Yeah, so I have a FASTQ report, another one, but I'm not sure if it's a messed up read, and I would love you to comment on every aspect of it and where like the mistake could happen.
Yeah, I mean, well, yeah, sure. We can stay online after the class.
Okay, sounds good.
Okay, so yeah, so that's it for FASTQ report. And one thing that kind of appears here is that you're just like, okay, where does this go? And the thing is, here we're trying a lot more to, I mean, we're trying to rule out errors more than we're trying to guarantee that our data is what it should be. If we have really big artifacts, then we're going to see it. If we have small artifacts, we might have a doubt on something, but then we can just keep going with the analysis, and maybe we're going to remove that one sample later. So yeah, there is like, essentially we're looking for an absence of bad patterns more than we're looking for specific patterns. Yeah, if you don't see anything that is suspicious, and it's pretty that your data is good. So it's kind of an absence of problems that we're looking for. Yes. Any question on this exercise? Okay. Did you all manage to transfer files to your laptop from the cluster? That's also like one of the goals of the exercise actually, is to have you transfer files back into the cluster. Getting comfortable with that.
I used like a graphical client and just logged in with the same credential.
Yeah, no. So if you manage to do it through CyberDuck, I think that's the best. I'm using it, but SCP is also fine. I mean, anyway, it's fine as long as you have a way to transfer files back into the cluster so that you're not stuck with not being able to look at your files here or there. Or it shouldn't be an issue that the files are on the cluster versus your laptop. And the way I function is everything big files is on the cluster, just because I don't know, my home connections maybe 1 megabyte second or something for like a stable connection. And so that's, if we have 50 gig of data, that's just not going to work out. So everything that's big file, big computation is on the cluster, and then visualization, I tend to work with my computer a lot more. Yeah. So download like summary statistics files and visualize what I have on hand. Okay, next exercise, we're pretty going to start now, but not finish. We'll see how that goes. It wasn't in the, I think it's become better since it's in the exercise PDF, but we'll see. Okay, there's a small issue with PDF.
Can I have a question? The trigonometric or cut at depth, which one you advise for and what the difference?
I think functionally they're equivalent in terms of like the quality of results you're going to get. I'm using trigonometric just out of habit. Both are very highly used. And again, these step, I think, I mean, it is always good to include a cleaning step like this, kind of removing the reads that are bad. It removes a bit of noise for sure, but at the same time, this was a lot more important 10 years ago when Illumina reads where the quality was a lot less, and it's really day and night from. I mean, current reads and reads from 10 years ago, it's day and night in terms of the quality. So 10 years ago, it was the really important step too. It was making a huge difference. Nowadays, it just removes a few reads, trims things a little bit, cleans up. I mean, it removes maybe a bit of noise, and still, it's an easy step to perform. It's not risky in terms of bias, so it's just good to still have it. But even not including it is not at all. I mean, not having a step at all is not going to make a huge difference in your output, and different software are going to perform similarly. Yeah, so I mentioned a couple of times that often in bioinformatics we have several alternatives, and many times you can just pick any of the widely used ones. And you can always like dive into the literature, try to dig the small differences and optimize, but it's a lot of time. I don't think it's in most cases worth it. It's really definitely not worth it trying to re-benchmark everything with your data. That's something you should not be doing. Really avoiding having, benchmarking several methods yourself. Essentially going for methods that have been shown to work for a range of data, and kind of relying on that. Some methods, I'm going to mention a couple, like the top hat software. That's kind of a bit outdated. It's not going to change your results entirely, but some software have been shown as being a bit outdated. It is, so what I mean is it's still worth looking at like reviews on the different software solutions for this or that problem. Oh, and one thing, since it comes up in the exercise, this data set, I haven't talked at all about paired end reads, and that's because this data set is single end. And so we don't have to care about any of the paired end stuff here. And so this exercise, so the goal is the immediate goal is to run this Tremomatic cleaning step. But the more generally, a lot of times in bioinformatics, you're just going to find a command either online or maybe someone like a colleague is going to give you a series of commands, and you're going to end up having to modify these commands. And so this is like a typical case, right? You see the Tremomatic web page, and actually the clearest way description of how you should be using the software is just this example that they're giving. And then you have to adapt it to your own needs. So that's in general, like obtaining a complete command and having to tinker it to adapt it to your own needs is something very common in bioinformatics. And then you kind of, the first thing you do is just figure out what the command's doing, why people use this and that option, and what their goal was for this and that, and then you can just like adapt it depending on what you want to do. Anyone wants to kind of walk us through the command line, or what they've understood of the command line? And so this command, like in the PDF, it's like just to clarify so that everyone's is really clear with it. In the PDF, it's like has these small like new line symbols, but actually in this case that means like the line continues on the next line. In the source document for the PDF, it's just one big line. Also, having documentations that's a bit all over the place, like for traumatic is quite typical for bioinformatics.
So mine's still running, so I don't know if it's working.
Oh,
or
your
not,
traumatic?
Yeah, I can show the steps that I took to create the command. I just don't know if it was correct.
It probably was. Let's see.
Oh, it just said completed successfully. Hooray. Let me just share real quick.
So usually when a program starts running for more than a few seconds, it's already, that's kind of like the first step for having the command line to work. It means that, like, for instance, the files are not missing, and then there's no typo, no obvious typo in the file. Sometimes you still end up with errors or weird things. But if it's running for more than a couple of seconds, it's likely that it's going to go to completion. It doesn't mean it's going to go to completion without errors or without doing the thing you wanted to do, but usually it means it's going to go to completion.
I guess the first thing that I did was I loaded the Java module. So that was module load Java. And that just returns, it doesn't say if it worked or not, but I guess it assumes that it worked. And then I copied and pasted this command here into, I use Notepad++. And I turned this into like one true single line command first.
Oh, when you copy paste, it's not? Okay.
Yeah, I don't know. I'm in on Windows and
Yep.
I don't know, maybe something about how I copied it, but when I copied it directly from this PDF over here, it had two new lines, and that kind of messed me up initially. And then I had to change all these things that you included here. So the correct path to the program has to include this here. So I copied this and replaced this command with the proper thing. And again, that kind of breaks it into two lines. So I had to put it back together there.
Yes. So that's a PDF, and I can't really do much against a PDF. But this is the reason why I insisted, like in the email that some of you have received, and then in the exercise as well. What you really don't want to do for any code-based applications is working in Word, or working in PowerPoint, or working in like Google Docs. It's just going to introduce weird characters all over the place, like new lines that you hadn't intended. It's going to like convert your quotes. It's going to convert your hyphens to things that are not quotes or not hyphens in the language of a computer, of a terminal. And you're just going to start seeing weird errors all over the place. Yeah, that was in parentheses.
And then I had to put this in as the correct input file. So I'm just hitting Control-C on there. But this also needs a bit of a change to the directory. So I had to put this in here, and then day one as the correct folder. And then I think I could just specifically set the output to the thing. I just picked the one that you recommended as a file name, but I guess you could choose anything here. So I replaced the default with that output name. The next step, I didn't really fully understand what you meant here, so I just ended up deleting the Illumina clip part. So I just get rid of that single command. And then I filled in the numerical values that you recommended. So for minimum leading and trailing quantities of 20, I changed this to 20 and that to 20. And then I changed 20 over a 5 base pair window. I just sort of assumed that the larger number should remain the larger number. So I changed that to 20 over here and this to 5. So I guess I can just do that like 20, 20, 5, 20, and then you wrote to make the minimum length some, more than half fraction, whatever. So we're doing, since we're dealing with sequences that are like in the 70s, you recommended 60. And I think that completes the command. And so then now I selected this entire thing and copied it and went back to my shell and copied it in. And I hope it doesn't hurt anything to run it again. But yeah, it's, oh, sorry, I forgot to add the threads. But it looks like it, even without that thread command, it's automatically using single thread.
Oh yeah, the thread. So the thread is I don't know. It's like sometimes I've seen it behave weirdly. If it just uses the single thread, sometime in the cluster, depending on which computer within the cluster you are, it's just going to try using like, I don't know, like 12 threads, and it's just going to create problems. But the thread, anytime you run a program with one or multiple threads, if the program is properly programmed, it should not make any difference. It just should speed up things when you use multiple processors. And thread and processor are kind of the same idea. Can you do ls? Yeah. So one thing I can mention here, and this is exactly how you're supposed to run the program, and you modify the right things. One thing you can comment on is how you needed to modify the input file to have the path, like specify the full path to the input file. So like after thread 33. Yeah, so that here. And so here really, like the program, what it expects is a path here. And whenever a program is asking for a file name, it's actually expecting a path. And the file name is really just the last bit of the path.
Sorry, I didn't mean to interrupt you. A mistake that I made, and I was unfortunately in this folder, programs, which I should not have been. I should have been in my own folder. So when I ran when I just put the output file in as just a file name, it dropped the output here into this other folder. And if I had specified like a full path, I think to my own section, it wouldn't have done that.
So actually, I can take the sharing here.
Yeah, I'll stop.
Where's my? Yeah, can you see this? Yeah. So one program, so for paths, one program that we can, that I really recommend you use, like if let's say I was running fastqc, and then okay. And so I've tried running this command here, and it failed. It told me like p10k or rep1 fastq.gz does not exist, which is true, because so the actual reason here is this is not a capital O, this is a lowercase o. And so I mean I could fix this and like make the command work, but the point was to show you. Let's say I haven't seen this, and I'm just like, this is weird. This entire command here, which here is very simple, but this might just be part of a much more complex command like the one for Trigonometric, right? And then the error is this one file didn't exist, and clearly that was not my intention that the file didn't exist, right? And so whenever you have an error of this type, the first thing you can do really is just confirm that the program is actually telling you that something that makes sense. And so the way you can do this is just take the path and then put it in ls, and then ls is also in principle going to tell you, yeah, this doesn't exist. And this is just the first step of confirming that this is the cause why your bigger program or bigger command line is not working, because then you have isolated the error and you can fix it. And then once you fix it with ls, then you can apply the same fix to the bigger command line and fix the bigger command line. And usually that's the fastest way to fix a big command line. Anyway, yeah, so what I wanted to say is this is, so ls takes paths. And so here we're showing this one file. This is just our path. And what I want to say is this just file name here is a path. And it's just a file name, it's just a file name in the path. It's actually equivalent. It's like entirely equivalent to having this // here. So just not adding any directory information is the same as saying this is from, I'm looking for something in the current directory. And the current directory in this case is just the day one directory where I am. So when in the Trimomatic line you specified, let's say I'm not in the day one directory, then I can't do the, like this is not going to work, right? Because this is a relative path and it's looking for this file in the current directory, and of course I'm in one directory above where I should be. So the file does not exist of the given path. And if I do this one without the // it's going to say the same thing. And really what it's asking for is for me to say, okay, I'm just going to start from the current directory, then I'm going to go into day one, and then that's where the file is. And again, I don't need the // at the beginning. It's like it's just for, I mean, it's a bit more like verbose. To have the // takes 2 more characters. It may help with comprehension, or maybe it's just like bloating the command line, but they're both equivalent. So whenever we're talking about paths and whenever a program expects paths, if you're not, and in contrast, like if I do this tilde here, it's actually going to be expanded into all of this, right? So this is my home directory, right? The tilde means home directory, and this is my home directory. And a path that starts with a tilde is a path that starts with a /. So it's an absolute path, right? So this file is going to be found regardless of which directory I'm in. So if I'm in the programs directory, it's going to be found all the same. And so that was the issue with why you had to specify the path. Is that, and regardless of what you're doing, you're specifying a path because you're giving the file name, and the file name is a path. But the first time it didn't work out because you were in not in the day one directory. And so for paths, you always have kind of 2 options: is changing your current directory or giving full paths. And the thing is, by giving full paths just for the input file, then you fix the input the path for the input file, but then the output file was just the current directory. I mean, in the current directory, right? So by fixing the problem by giving an input path, a full path for the input file, you cannot introduce the problem that the output file was not in the right directory. Does this make sense? Anyway, it's very normal. Paths issues are like normal, just like the everyday thing in Unix. But yeah, really working with making sure, yeah, just paying attention to the current directory is one important thing. But yeah, no, that was otherwise. Yeah, that was a command you're supposed to use. And so you can just modify it for the second sample, just making sure you, yeah, the first time you kind of tinkered quite a bit with the command to make it work, and the second time it should just be the sample name that changes. Oh, one thing I can mention regarding rerunning it for another sample is we're going to get into the same sort of issues actually, where it's very easy to mix up kind of what we're doing with the input file and what we're doing with the output file. And so, for instance, what may happen is if I go back to my terminal, and then, yeah, I see what you mean. Yeah. So in this case, I'm just having this like very fancy symbols here. Then, just very quickly changing the line. Okay, so this is so let's say this is our first command, and it's going to take some time to just run. And then essentially this is working on the first sample, right? P10ko rep1.fastq.gz. And so now we want to change this sample, right? So instead of P10ko rep1, we want to work on P10 rep1. So that other sample which is not in the KO group. And so we've modified our command line, and then we run it again. And what just happened? Did anyone see the problem?
You made the output the knockout file name.
Yeah. So when I changed the command line,
does it like overwrite?
I actually had two places to change, right? I changed the input file. I just like took one of the names, and so I changed this. So it's actually working on the data from the first sample, but the output file is still the second sample. It's still the input file is the second sample, but the output file is still that of the first sample. And so in that case, it's not really going to be very serious, right? Because I'm just going to realize I'm now missing the file for the second sample, and something went wrong, and I may just start again, like remove all the files, start again. Or I could do something else which is just, I'm missing the second file. So start again with the command line, copying and pasting everything, and then updating the file to work on the second sample. But then if I did this, what would have happened is that I've actually duplicated the second sample and removed the first one. Just because I mixed up I edited things by hand, right? So this is just one sort of human errors you can make, and I've pre-made some human errors like this, and then you probably would realize later on in the pipeline that two samples are strangely identical, like exact same number of reads, exact same gene expressions, and which is just not possible, and you'd have to like go back all the way and redo everything. So it's better if we don't like run into these problems. And so one way we can do this is actually to use shell variables like this. And now we have stored our sample name inside this sample variable, which is really just a shortcut name for getting to the sample we're currently working on. And so I can take my command line like this, and in this case, this is a command line with like the two different samples. But actually, the idea is that we just have like this blank thing here, or we just have these two places in the command line that should have the same value, right? And so this idea of shell variables, that's the tool that's going to let us change these two places at the same time, and then we don't have the risk for human error anymore. And this is how we can use our shell variable with $ and then the name of the variable. Okay, and now we have those two places that have the same sample name. And one way I sometimes use, and it's like 4:30 soon. One way I sometimes use to check my commands like this, to check that everything is in order, especially when you start having variables and everything, is just to add echo at the beginning, right? So it's not going to run the command, it's just going to echo the command itself. And so we get this. So this is what the command is actually going to look like for the shell, for the cluster. And here my variable has been replaced, and I do have the same command in both cases. And the good thing is, like, if now I'm changing the sample I'm working on, so instead of p10 rep 1, like here, I was saying here, I'm working on sample p10 rep 1, and I'm storing this is the value of the sample variable. And I can change this, and if I look at my command again, now it's changed, and it's changed in both places. And that's why I really just sometimes using this, having the variable definition, a single variable definition where I'm writing the sample in a single place, is really helping avoid these human errors. And also I'll talk more about variables tomorrow, and we're going to like keep building on this. But yeah, okay. Yeah. See you tomorrow, everyone. And yeah, have a good night. Yeah, I can stay for a few minutes for sure.